In this mini tutorial we're going to consider two humoral components of the innate immune system, these being complement and interferon. And these are two extremely important systems which are evolutionarily ancient and are part of the first line of defence when a pathogen breaches one of the immunological barriers. Now, complement, we'll consider in a bit more detail soon, is essentially a system comprising about 30 individual proteins which during most of the time are in an unactivated state, but in the presence of pathogens the complement proteins can become activated and we can get some very specific responses against invading pathogens. They're called complement, by the way, because when they were first discovered they were said to complement the role of antibodies. Interferon, there are a number of different types of interferon, and what the interferons do is they are involved with interfering with viral replication. And we'll talk about those in a bit more detail as well. So let's think about the complement system in a little bit more detail. Now, effectively there are two different ways in which you can activate the complement system. And just to get this straight in your heads, all of the 30 proteins in the complement system are circulating in the blood. So these are all present in the blood, just waiting to be activated by a pathogen. The first pathway that we can activate complement by is known as the classical pathway. And the second pathway we can activate complement by is called the alternative pathway. So there are two different routes whereby we can activate this system of 30 pathogen-killing proteins. Now, the classical pathway is typically activated by the presence of antigen bound to antibody. Alright? These so-called antigen-antibody complexes. So if components of the classical arm of the complement system see antigen bound to antibody, then they will activate the classical pathway. The alternative pathway of the complement system is typically activated by pathogens themselves. Specifically, it is activated by pathogen surfaces. So these would be bacterial cell walls or fungal cell walls, for example. But regardless of the way in which the complement system is activated, the effect is the same, and that is activation of the rest of the components of the system. So the rest of the proteins in the system get activated, whether it's via the classical or the alternate pathway. And upon activation, there are three different effectors which can be switched on. The first effector system, which can occur upon activation of the complement cascade, is recruitment of immune cells. So various components of the complement system, once they are activated, can actually attract immune cells and make immune cells come to the site of the infection. The second effect which activated complement can have is known as opsonization. Opsonization. And essentially what opsonization is it leads to flagging up of a pathogen to phagocytes. So opsonization is the process whereby we're advertising these pathogens to phagocytes in the immune system. We're making them easier for phagocytes to take up. And opsonins are molecules which stick very well to pathogens and make them obvious to phagocytic cells. The third effector function of the complement cascade is direct killing of pathogens. And the way that complement typically kills pathogens, usually bacteria in this case, is by forming holes in the bacterial cell wall. And complement can generate what's known as a membrane attack complex, which is a big molecular complex which can create big holes in the cell walls of pathogens. And if you make holes in the cell wall, you lead to the death of the pathogen because it can't regulate its internal environment. So, of the complement cascade, and you should do some more reading about that on your own. Now, the second system I want you to think about is the interferon system. And there are various different types of interferon, but the types of interferon I want you to think about are interferons alpha and beta. And these interferons are released following viral infections. And essentially they do a number of different things. So imagine that we've got here a virally infected cell which is filled with viral particles here. The cell is about to die anyway. And what this cell does is it releases a whole load of interferon. And this interferon does a number of different things. One thing it does is if it gets to a neighbouring cell, then what the interferon does is it causes changes within the cellular metabolism to inhibit viral replication. So it's essentially a way of one cell signalling to another and telling that cell to shut down protein synthesis, for example, to prevent these viruses from replicating within that other cell. Another thing that interferon does is it activates macrophages. So it activates macrophages and other phagocytic cells. And what that does, of course, is it's stimulating the uptake of viral particles. So if we activate macrophages, those activated macrophages are going to start phagocytosing any viruses which are released by infected cells, as well as phagocytosing other virally infected cells, or debris left from when a virally infected cell lyses. So interferon can cause activation of macrophages. Additionally, interferon can stimulate natural killer cells, NK cells. And what natural killer cells are able to do is to come along and directly kill virally infected cells. You've all got direct experience of the effect of natural killer cells because, for example, when you get a sore throat having had a cold, one reason that you get this sore throat is because natural killer cells have come along and have started to kill the cells lining your throat. And in fact, a lot of the symptoms you get in the common cold are nothing to do with the virus itself. The symptoms are actually caused by the release of interferons. Okay, so I hope that's clear.